My name is Rüdiger Hell. I'm located in INF 360. You find me in the first floor, where my office has a beautiful view on the botanical garden. I've been in Heidelberg since 2003, and this is a major part of my life. My career started with my PhD, which I carried out at the University of Cologne. At that time, it was a must to change to the United States as a plant biologist. And this is what I did when I became a postdoc at the University of California, Berkeley, where I spent wonderful 2.5 years. However, in 1993, I returned to Germany to the Ruhr University Bochum, where I started a lab on my own and independently and carried out my habilitation. Directly after that, I moved to the Institute for Plant Genetics and Crop Plant Research in Gartersleben in the so-called Neue Bundesländer. And it was there where I received my call at the chair for plant molecular biology here in Heidelberg. First at the Heidelberg Institute for Plant Sciences, and since 10 years at the Center for Organismal Studies. Early on, I was interested in natural things, life, insects, butterflies, plants. Everything was of interest. But when I was still going to school, I did not obtain very answers, or at least not satisfying answers. And I decided to figure out things on my own. And when I learned that biology is a study subject, I decided to go for biology. After I completed my studies, I had to decide if I really wanted to go into a real job or into academics, which can be a job, but at that time I was not really fully aware of that. So I took decisions step after step. After my studies, I decided for a PhD. After my PhD, I decided for habilitation. And honestly, after that, there was no way back. This stepwise development of my career always went along with having a plan B in mind, because becoming an academic biologist was a very difficult endeavor, and the chances, honestly, were very dim. So I always tried to cultivate in my mind which other options I have. In the beginning, I simply thought I will do anything else where I can make a living. But soon I learned that I'm very much interested in communication, and especially in communication of science. And in a way, teaching is also communication. But I could imagine also to be active in publishing or featuring science aspects for print media or live media. So this was what I always had in mind if anything goes wrong with my academic career. I'm teaching in plant physiology and plant biotechnology. I'm active in the third semester with plant physiology, especially stress physiology. I'm active in plant molecular biology and plant biotechnology, where I'm teaching transgenic plants and the comparison to conventional plant production. I'm offering seminars, introduction into biotechnology, synthetic biology, and aspects of plant adaptation to stress. We are also offering practical courses where my emphasis is really to provide the opportunity to work with the students' own hands and not to digest things that have been prepared to a very high level. It's the working with your own hands and the understanding what you're doing that is the premise of these practicals. Teaching is, first of all, the way to stay in contact with young people. And it's the big chance to stir the enthusiasm for the subject that I love so much. Biology is really great. I wouldn't have done anything else, and I do not regret any decision into this subject. So if I manage to raise some of this enthusiasm, to confer it to the students and motivate them, and then make them find their own way, because every person is different, that is really the greatest fun. I'm trying to understand how plants work. I find it fascinating that they're able to convert inorganic substances into organic substances, and how plants gain their energy and provide the basis for development and adaptation is investigated with respect to sensor kinases and the conversion of single metabolites into large molecules. making up in the end the organism. We're using a very broad range of techniques to achieve this goal of understanding the whole organism. Of course, it starts at the molecular level. We are cooperating with crystallization platforms, but it ranges also into flux analysis of metabolites using labeled substances. We're using a lot of confocal microscopy to understand live cell processes, and we're doing a lot of genetics, manipulating plant metabolism and investigating the changes that we induce in order to give us the answers that we need to understand the cell and the entire individual. I'm mainly using Arabidopsis in this work because it's the best investigated plant and it offers a huge toolbox. But we always like to have one or two crops to work with, for instance, rice or maize. This is much more complicated, but of course, with respect to human nutrition, very rewarding if you can achieve any progress with these crops. In these projects with Arabidopsis or maize or rice, we are mainly looking into the role of sulfur metabolism. Sulfur metabolism is one of the basic primary pathways and controlling a lot of internal processes. It is basically for us a tool to investigate how plants sense their environment, in this case the availability of sulfur, and how they translate the availability or the lack of sulfur into their growth behavior, their growth rate, and the differentiation of root and shoot. A second topic in this context is water deficit, probably the most limiting factor for crop production worldwide. How plants sense the water deficit and how they react in order to keep the water inside is intricately connected, in fact, to sulfur metabolism. So I have two projects that are basically joining into one big question. All these investigations, of course, cannot proceed without failures. Honestly, most of our hypotheses are wrong. And yes, we experience a lot of disproving our hypotheses and eventually approximating the right answers. That usually works, but it is a long way. I believe science cannot work without false hypotheses in order to find the truth. This way of raising hypotheses, hypothesis-driven approaches, but also, of course, unbiased approaches like omics technologies, leads to many unexpected discoveries. And that's actually a big fun part if you find something that you did not anticipate and did not integrate yet into a hypothesis. A very good example is that when we investigated a protein complex that is actually responsible for the synthesis of cysteine, we found out that this goes along with resistance against water deficit or drought stress. And that is something we are now following up in our projects. The broad array of techniques and different questions that we are raising requires a lot of scientists and hands helping. So we're always open for practical students, for Hiwis, but we're also offering bachelor and master projects. I've seen a lot of students in these years as an active research scientist, and I always try to give the recommendation of being very open. Try to grab as much information and knowledge that you can. Carrying out your studies is the opportunity in life to learn as much as possible. And the more you know, the better are your decisions for your own career and for your life. So don't focus early on, even if you already know what you want to do in the future, but try to study openly also other subjects. And they always are interesting and provide surprises, and eventually this is the basis for the best decisions. Most important qualities for a scientist are a natural curiosity combined with motivation. Everything else can be trained and learned. But if there is a drive in a person where you can really see there is curiosity trying to answer questions, these are excellent prerequisites to become a scientist. A real high proportion of funding without administrative hurdles would of course be a great gift. And that's what we are always trying to get actually. What I would like to set up is a research project where scientists can really develop in terms of having sufficient time to accomplish a project and develop it to a point where we can produce knowledge that eventually ends up in a textbook. So a young researcher should have the opportunity, without worrying about time limitation, contract limitation, or funds, and really answer questions. That would be great to set up a team like this with a good proportion of funding.